Okay, well, I'm Miranda Fowles. I worked with Seabird Scientific, which is an oceanographic instrument company, on the project Yaquina Bay OH monitoring. The beginning of change in the bay is what I called it. So we had three main goals and kind of questions for this project to examine, investigate, detect, and analyze. Our first main question is to examine short-term variations, and short-term variations is because I'm only here for 10 weeks, but in oxygen, pH, temperature, and these are influenced by many factors such as solar radiation, tidal movements, and upwelling. Our second main idea is to investigate the presence of oceanographic signals, particularly in terms of oxygen and pH within the coastal of Yaquina Bay. And our last main idea is to detect and analyze instances of low oxygen, which is considered to be hypoxia, or low pH, which is acidification, that could threaten local marine species. And my main goal of the project was I was coding with RStudio to interact with graphs that compared the oceanographic instruments in the climate monitoring station, which is CMS, to see if the different biogeochemical measurements are comparable or if adjustments need to be made. Okay, so this is the climate monitoring station. Yeah, and this provides long-term time series data on physical, chemical, and biological properties with a rising threat of ocean acidification and hypoxia, which is OAH. There is a need to understand how its combined effects and annual timing impact certain marine ecosystems. And the CMS and the OAH Oregon Action Plan will allow for ocean monitoring and research to inform state-level mitigation and adaptation strategies. So we can see on here, this is the climate monitoring station. On the left-hand side is actually just behind us on Hatfield's campus. And the big white arrow is pointing to the pump house and the pier where the station is located. The middle picture is the station, like the house, and the right picture is inside of the building. And you can kind of see that there's a metal cage. I'll talk about that a bit later. That's kind of the main whole climate monitoring station, and that's lifted above. Normally it's in the ocean, but and in the cage, there's four instruments. Our first instrument is the CFOX V2 Ocean CTD, and CTD stands for conductivity, temperature, and depth, which is measured by pressure. This collects depth, temperature, salinity, pH, and dissolved oxygen. Our second instrument is the SUNA V2 nitrate. This collects nitrate concentration from UV absorption spectra. And our third instrument is the ECO triplet W, which collects chlorophyll A concentration estimated from a fluorescence of about 470 nanometers. And it also collects dissolved organic matter estimated from fluorescence and particulate backscattering. These first three instruments are Seabird Scientific's instrument, and our last instrument on the cage is from another oceanographic instrument company called YSI, and it's called the XO2 sonde. And this instrument collects depth, temperature, salinity, pH, dissolved oxygen, turbidity, and total algae. So how we get our data from the instruments to our computers is through the CMS data logger, which is a Campbell Scientific data logger. This logs all the variables from these instruments, and it's supported by access from OSU, so how we process it. So this is basically the instruments take measurements of our variables, and it's sent to our computers, and that's how we can analyze and look at the data. So the EXO collects data every 15 minutes, and the other three instruments, the first three, collect data every 5 minutes. So so you can kind of see a bit more, I can point them out. So you have here this gray one, this is the CFOX. It's a little hard to see, but behind that's the EXO, the blue one. You can kind of see it a bit better here. And then this silver one is the SUNA, and this little black one down here is the ECO. So this is how it's kind of made in the cage. And then you can see how we deploy it. On the left-hand side is me. That's how you get it up and down into the water. You press a button and it pulls it up, and you press another button and it puts it down into the water. The middle picture, it's kind of like the top orientation. You can see the four different instruments, kind of how they're located, separated. And the far right is that's the hole that we put it down into the water. And it is the cage is deployed 1 meter above the seafloor. right there. Okay, so this is kind of the raw data to the graphs. The left-hand picture, you can see it's an example of the EXO. So that's one of the instruments, and it shows us our timestamps, and then it goes into different signals such as pH, salinity, conductivity, and that's just how you see the first-hand data. I put this into an Excel file, so that's in Excel. And then you go to the right, which is how I'm um, looking at the data. I've developed scripts to change the raw data into graphs that are more readable, and this is in R Studio, so you can kind of see left is like my code, and then it turns into a graph, so more people can understand. Um, this is kind of a side note, but another thing I've been working on with my mentor is an experimental box. The purpose of the experimental box is to provide power and data connections for a variety of instruments to deploy to support experiments, instrument innovation, and instrumental comparisons. This allows for like other organizations or PIs to connect their instrument and they can analyze their own data and just like a way for people to collaborate. And this box is going to be put into the climate monitoring station, like the house on the pier. We kind of put together, you can see the left-hand picture, that's what's going to go into the box on the right-hand side. And we put together the left hand. Some of the parts that are in it are the uninterruptible power supply, which like provides continuity and power delivery and data collection during short interruptions. We also have a power supply which powers the instruments. And then we have a serial port server on there too, which collects data from the 16 instruments. And you can kind of see that it's the gray on the top and the 16 different holes. That's where you can plug in an instrument. We also have and internet accessible relays, and this powers on and off the instruments or experimental treatments according to a schedule that you have. Terminal blocks to attach the cable and an Ethernet switch, which connects it to internet accessible devices. Going back to kind of like the main part about the instruments, the main goal I was working towards was comparing the data collected between the CFOX and the YSI XO2 SOND. And this is because the YSI complies with EPA's protocols, so they have a long time series of local measurements with it, meaning we can validate the data from the CFOX. And that shows that like the XO is probably the more reliable data, so we can compare the CFOX to the XO and make sure that the two instruments are lined up. So going into this, this is my first graph. This is temperature and pressure versus time with the CFOX and the XO. On the top graph, you can see the light blue is the CFOX temperature and the red is the XO temperature. Temperature measured in Celsius, and the bottom is the CFOX pressure in the green. We're looking at pressure because this shows tidal signals, and signals are tidally driven. So this is a really good way to see like if the instruments are reading correctly because I'll zoom into like one tidal signal, but it helps us see if they're accurate. And you can see temperature is one of our most accurate signals because the red and the blue are very lined up, so both instruments are reading the same data. I'm gonna like zoom in. This is on one tidal cycle of the temperature, so just the last graph you saw. Now this is zoomed into one day. And you can see the tidal signal, high tide, low tide, the medium high tide. And this is like, it shows us because you can see on the top here where the temperature is cool and this is high tide, and that's because we're in a estuary in a bay. So high tide will have colder water because the ocean water is coming in, and that means it's colder. Low tide, it's warmer water because the ocean water is going out. In our next, I looked at dissolved oxygen with time and CFOX and XO2. Again, the XO is the red and the CFOX is light blue, and this is dissolved oxygen levels. The little greeny box thing is looking at hypoxia. Hypoxic waters are from 2 to 5 milligrams per liter, which is the units on this, so that's why the green box is there. And hypoxia is low dissolved oxygen conditions, and it's important to keep record of this because hypoxic conditions can stress out certain marine organisms. So we're really looking at this green, and it's important to have this green box because it can really show us, are we in hypoxic conditions or not? So you can see that the XO like kind of started in hypoxic and then maybe left, and then that the CFOX is more in hypoxic. And yeah. Also, the XO reports in milligrams per liter, but the CFOX reports in milliliters per liter. So I had to convert the CFOX to the XO and change the units. We can see that they're not lined up. So this is one of our issues right now that we're trying to solve. Like, why isn't it the same data? Because you can see on the cage that the instruments are right next to each other, and the sensors are maybe only a few centimeters difference, but they should have very similar data. As you can see, they're not. So that's a next step. Um, the next graph is looking at pH versus time, with again, light blue being CFOX, red is XO. You can see that it's in the range of pH, like normal pH levels, but also again, it is not lined up. So that's another question. We're not sure why the pH is not matched up. It could be because the seabird CFOX is temperature and salinity corrected, whereas the XO is not. So these two measurements are not independent, so the same issue could be affecting both pH and salinity, which I will get into in the next slide. We also, another side note is that we use internal pH because that is said to be more accurate for estuary water. Um, our next slide is going into salinity. So again, you can see that the salinity is very different and we don't really know why, but it is in the range of ocean salinity, which is in the range of like low 30s. So that's correct, but they're very different. Salinity should be high at high tide and lower at low tide because in high tide, there's more ocean water, low tide, more estuary water. So you can kind of see the tidal cycles in the EXO specifically. It's going up and down, but the sea fox is a little just scattered randomly more. We think the salinity didn't line up because the calibration not being possibly accurate or the sensors are at slightly different heights, but that shouldn't make too much of a difference like we're seeing. But we know the XO is probably the more accurate because of the EPA and how I said earlier, and also that we ran a test with the CFOX and we noticed that the pump is reading old water instead of getting a tidal push through, which is how it normally should be reading the data. So that's something we've been working on. We've been like going out there and kind of fiddling with it, but those are some next steps too. And that is all I have. Thank you so much to all of my mentors, Eric, Reem, and Marnie for helping me out, and Cynthia, Mako, and yeah.